lectures. Last time we talked about how DNA replicates, what can go wrong with the replication, and how we can fix it. We're done with that. Now we move on. We're going to talk about gene expression. And what is the first part in expressing a gene? Yeah, we need to do transcription. The first part in gene expression is transcription. Okay, so and this is what we will focus about today. So we will be talking a lot about RNA, type of RNA, how we produce the RNA by transcription, how we process the RNA, and we're going to compare between prokaryotes and eukaryotes. What are eukaryotes? Human cells. Okay. Let us review the flow of genetic information. So who can remind us what is this diagram saying? Yes? I want to see hands. What is this is the central dogma of molecular biology. What does it tell us? Yes, you at the back, the very end, yes?
Tell us that to get a protein, you need first to unwind the DNA to get mRNA, then you translate it to get a protein.
Okay, so yeah, it doesn't talk here about unwinding, right? But this is extra thing. Yeah, thank you, but what you said is correct. So we need to make a protein, to express the gene, we need first to make RNA out of it, and this is called transcription. Then we make a protein, and this is called translation. It doesn't happen in one step. You cannot express the gene in one step. You have to do it in two steps, transcription and translation. Okay, if we look at the wording, transcription and translation, they're both talking about language. It's like you're doing something in language, right? Transcription and translation. In language, what is the difference between transcription and translation? When you transcribe something and when you translate something, what's the difference? Yes?
Translate is you change it from one language to another.
Yes?
Transcribe it is to write it down.
Okay, so basically writing it down in the same language, right? Excellent. And how does this apply here to gene expression? Yes? The same hand, yes.
When you transcribe it, you just make an exact same copy of the DNA, but when you translate it, you change it from DNA to RNA.
I didn't know the first part, but nice try. So the first part about language, it was absolutely correct. Who can fix the second part? How does this, transcription and translation apply to gene expression? Yes?
So you have the transcription, you can convert it to proteins.
Okay.
It's like a recipe that
Okay.
you take and make it proteins.
Okay, you're getting there. Okay, who else want to say something? Yes?
Transcription is like writing down the DNA code in form of mRNA. And that mRNA is translated into amino acids and into proteins.
Okay, but what does this have to do with the language? If you use the same analogy in language, yes?
So DNA and RNA are both like the same language as bases. And
Excellent.
then RNA and proteins, you go from bases to codons.
Yes.
Excellent, excellent. Zafulo? Okay, so if you understood that, you will understand and you will never forget what is transcription and what is translation. When you're transcribing DNA into RNA, you are copying, like your friend said, from one nucleotide sequence into another nucleotide sequence, right? Their DNA and RNA are both made of nucleotide. You're just copying the information from DNA into RNA, and we will see how this is copied today. When you're going from RNA to protein, you're going from the language of nucleotide, okay, because DNA and RNA are made from nucleotide, to a completely different language, which is what? Amino acids. So you need translation, and when you translate, you need to know the meaning, and this is the genetic code. Okay, so when you talk about translation next week with Dr. Ahmad, we have a new professor of biochemistry. He will start with you next week, and you will have really, I'm sure, a good time with him. So he will talk about translation next week with you. Anyway, so when we learn about translation, you will know that proteins are made of amino acids. So we're going from the language of nucleotides to the language of amino acids. So we need to have a code that will tell us how we translate. We need to know the meaning of the nucleotide in the amino acid language. Okay, so you need to have this genetic code, otherwise you cannot translate. Okay, if you don't know what's the meaning of, I don't know what, sugar, let's say in Arabic. I mean, sugar will always be sugar, unless someone tell you that, you know, this word has this meaning in that language, which is the genetic code here, you won't be able to do the translation. Okay? So this is the difference between transcription and translation. In transcription, we use the same language, the language of nucleotides. And translation, we're translating from nucleotide to amino acids. Okay? Clear? Clear?
Yes.
Okay, good. Okay, last time we talked about the purpose of having the transcription. So why we don't go from DNA directly to protein? Why do you think? You know, same hands, yes.
Maybe to avoid damage, like when the RNA comes damage, there is some technique for repairing, so the protein will be functional.
Okay, maybe, yes, Mu'ad.
There is a modification of the mRNA before
Yes.
translation.
Mm.
So the I think the introns or the exons are removed from the actual mRNA
Okay.
that's translated.
So yeah, we do modification, but what do you think is the ultimate reason why we just don't go directly from DNA to protein? Better.
Because the DNA can't leave the nucleus.
Exactly. The DNA is inside the nucleus. I got it right, Mu'az better this time, yeah? So I got it right with you and he got it right. So the DNA is inside the nucleus. It cannot leave the nucleus. So we want to send a message. So who's going to carry the message? It's the messenger RNA. It's going to leave the nucleus and go into the cytoplasm and meet the ribosome and it will be translated. Clear? Yes? Okay, good. You were asking me a lot about the structure of the chromosome. So this is our chromosome. It is packed, you know, with this DNA double-stranded sequence. And there are many genes. So on each one chromosome, there will be many genes, okay? The new information today that I will tell you is that our gene is made of exon and introns. What is the difference between exon and intron? Yes?
Exons are the ones that are expressed, introns are not.
Exons are?
Exons are expressed,
Excellent.
introns are not.
Excellent. So exon, okay, they are expressed. It's easy to remember that exon expressed. Introns are like intervening sequence, okay? So they are between the exons and they are not going to be expressed. So we don't use the information, the sequence, the DNA sequence in the intron, we are not going to use it to make a protein, okay? So this is the difference between the exon and the intron. So the other word for expressed is coding. So exons, they contain the coding sequence, the one that we use to make the genetic code, you know, the code of from which we make amino acids. The introns are non-coding, okay? The size of genes varies. We can have small genes, big genes, and also the number of exons each gene have. So on average, a gene can have 7 to 10 exons, and they are spread over 10 to 16 kilobase. So, you know, variable size of genes. Okay? Because we're talking about how we make RNA, so we know that DNA is translated into messenger RNA, but is messenger RNA the only type of RNA inside the cell? No. But messenger RNA is the only one that is going to be translated, okay? The only one that we will use to make proteins. But we have other type of RNA that does different function or do different function inside the cells, and these are, we have the ribosomal RNA. From the name, ribosomal RNA, what does it do?
Thanks for
It forms the ribosomes. Okay, so ribosomal RNA to form the ribosomes, and it is the most abundant type of RNA. The second most abundant is tRNA or transfer RNA. Okay, you will learn about it next week. What does the transfer RNA do?
Transfer amino acids to
Yeah, it's going to transfer amino acids to be synthesized as proteins. Okay, so it's needed for protein synthesis and the messenger RNA that we'll talk about in detail today. Okay, so it is the one that carries the information to be used to make protein. Okay, it's the only one that's going to be translated, and the population or the different type of messenger RNA we have in the cell, they are of different size. Why they have different size? Because depending on the gene, we have big genes, small genes. So each one gene, although the DNA molecule is like one large molecule that make up the chromosome, the gene, they have various lengths, and from these genes we're going to get various lengths messenger RNA. Okay, and also once we make the messenger RNA, we do a little bit of processing to it. So we will have, processed, unprocessed messenger RNA, okay? So we will have a heterogeneous, so a different population of messenger RNA, okay? So we said messenger RNA, they vary in length because the gene vary in length, okay, this is one. And the one that we get to process, so that the one that is not final, the one that's not going to be used to be translated, is actually what we call heterogeneous nuclear RNA. So what is the heterogeneous nuclear RNA? It's basically the messenger RNA that has been transcribed and not yet processed, okay? What is processing? We will learn about it today, okay? So basically, heterogeneous nuclear RNA is a premature messenger RNA, a messenger RNA that has not been processed yet, okay? Small nuclear RNA is actually important for removal of introns, and there is some pathology associated with it, and we'll talk about it later today, okay? Small nuclear RNA. The micro or miRNA, okay, this is not a spelling mistake, there is an I here, it's not messenger RNA. This is the most newly discovered type of RNA. It's actually the one that turn on and turn off gene, okay? And it is, again, non-coding, so we don't use it to make proteins. They are very short, about 22 nucleotides, so they're very short, okay? And they can control gene expression, usually during development. So these are the microRNA, okay? These are the newly discovered type of RNA. And we have the ribozymes. So as the name indicates, zymes, so they have enzymatic activity. And these are RNA molecules who have enzymatic properties. And they are found both in pro and eukaryotes. Okay, so this is summarized here. Okay, so this is a table summarizing the different types of messenger RNA. So as you can, the different types of RNA. Messenger RNA are the only one that code for proteins. This is the one that make ribosomes. The miRNA control gene expression. Transfer RNA for translation. And then we have small nuclear RNA, which are important in things like RNA splicing. Yes?
Doctor, in the microRNA, it says during development. Is it during normal development or embryonic?
It could be both, okay? It's for both, because it can control gene expression, okay, later in life also. But anyway, this is very new field, and it's like at the postgraduate level, so you don't need to worry about it. Okay, so now we have introduced the type of RNA, but today we will only talk about two types, the messenger RNA and the premature or the pre-messenger RNA. That's the only two types that we will cover in our lecture today, because we are going to be talking about these two, transcription and processing of RNA. These are the two topics that you will cover next week, translation and post-translational modification. So this is going to be covered in the protein synthesis lecture next week. Today and tomorrow we'll cover these two. Okay. When we studied DNA replication, what did we need to make DNA, to copy DNA before the cell divide? What are the machinery needed?
DNA polymerase.
DNA polymerase,
Primer.
helicase,
Primers.
the binding protein that opened kept the helix open, primase, RNA primer, binding protein, yeah, I mentioned them, and ligase to merge the Okazaki fragment, okay? So let us see here what are we going to need to copy or to transcribe DNA into RNA. And also you need to remember that, you know, in replication we had 2 strands, right? And when we replicated the DNA, how many strands we made? When you copy the DNA for DNA replication,
4 strands, 2 double strands.
yeah, of course, the cell need to divide. We're not going to give it half a strand for, you know, we need to copy exactly the same amount. So DNA replication, we go from double-stranded DNA to double-stranded DNA. And both strands are copied, right? So we don't have one that is copied, one that is not copied, right? This is in replication. Last week, we're copying the DNA before the cell divide. So we need to copy everything, right? Now, here, we're going from double-stranded DNA to, what is the RNA, double or single?
Single.
Single-stranded. Okay, so, are we going to copy 1 or 2?
1.
2?
1.
Okay, we have to choose 1 only, because we're making single-stranded RNA, so we're going to choose 1 strand to this side. So which one do we choose?
3 to 5.
3 to 5, 5 to 3?
3 to 5.
Let us see which one we will choose, and we'll answer this question later. Okay, now let us look at the machinery. Okay, so we are making RNA. So RNA is a polymer of nucleotides. So what will be the enzyme that we use to make RNA? RNA polymerase. Okay, so this is the RNA polymerase, that blue thingy here. Okay, so what does it do? It searches for initiation site. So what's initiation site? This is where you're going to start copying. But we have double-stranded. How are we going to copy double-stranded DNA? Okay, the polymerase itself now will unwind a short stretch of double-stranded DNA to produce a single-stranded template. So you see, we don't have a helicase here. The polymerase itself will open the double strand a little bit at a time. Okay? After it searches for the initiation site where it's going to start copying, it will select the correct. So for making DNA, we needed dNTP. Here we are going to use ribonucleotide, right? Because we are making RNA, so we need ribonucleotide, not deoxyribonucleotide, right? Okay, so we don't have dNTP, we have NTP, okay? Not deoxy, because the sugar in the RNA is ribose, not deoxyribose. So they are called ribonucleotide. And how we are going to link them? So in DNA, what's the bond that links one nucleotide to another? What was the bond? Phosphodiester bond. So here the same thing, we have phosphodiester bond because they are actually polymers of nucleotide. RNA and DNA are polymers of nucleotides. So they're both linked by phosphodiester bond. So it starts at the initiation site, open the double helix, select which strand to copy it into RNA, and then detect the termination signal. So we need to have a start and a stop signal where we end the transcription. Okay? Okay, so this is the polymerase at work. This is the double-stranded DNA. This is the opened double helix. Okay? And that blue thing that is being made is the RNA that is exiting the polymerase. Okay? And we are using ribonucleoside triphosphate. Okay? They are not deoxy, they are ribonucleoside. Okay. We'll talk about about the direction of the transcription and the synthesis of the newly synthesized RNA later. Okay, here we don't have helicase, we don't have single-stranded binding protein, but we need something to guide the RNA polymerase to tell it where to start. Okay, so this is the start of transcription. How do we know that this is the start of transcription? Because the RNA polymerase will search for a promoter sequence. So this data box, it means there is TA repeated. Okay, we call it data box. This is a part usually of a promoter region. So what is the promoter region? What? Yes?
The region before the gene that will be transcribed?
Yes, so it is the part of the gene, part of the DNA sequence that will tell us we should start here. So it is the part just before the start. So the RNA polymerase will be looking, this is the RNA polymerase, it will be searching the gene for the promoter region, okay? This promoter region will bind to something called transcription factors. So we need these transcription factors. We will learn more about them as we go in the course. So what are these transcription factors? They are basically proteins that bind to the promoter region and will allow the binding of the polymerase, to the promoter. So the RNA polymerase will bind to the promoter with the help of the transcription factor. Okay? So the promoter, they are just before the start of the transcription. Okay? They are sequenced in the DNA that will tell the polymerase, come here, you have to start transcribing here. Okay? There is specific sequence that is recognized by the polymerase to initiate the transcription. To this promoter region, we have some proteins called transcription factor that bind to the promoter and will help the RNA polymerase also bind to the promoter. Okay, so now we have, once the RNA polymerase bind to the transcription factor and the promoter, transcription can occur and you will start making RNA. Okay? So we will explain this as we go in more detail. We said that transcription, we go from double-stranded DNA to single-stranded RNA. So SS is single-stranded RNA. How we go from 2 strands to 1 strand? Only one strand of the molecule, of the DNA molecule, is copied, and this is going to be called the template strand. Okay, so the strand that we are going to copy is actually the template. We call it the template. And when we make the RNA product is going to be complementary and antiparallel. The same like when we made the DNA. Remember in DNA replication, the newly formed DNA, each strand was antiparallel and complementary. So here the RNA that we make is going to be antiparallel and complementary to the template strand. Okay? So, which one is the template strand? It's determined by the promoter and the terminator. So the RNA polymerase will recognize the start signal, which is the promoter, or what we call just before the initiation site, and it's going to recognize the stop signal for each of the many genes in our body. Okay? So which one is going to be the template? Which one is going to be the non-template? It depends on the gene. Okay, so it depends on the location of promoter and terminator. So let's pretend that this is one part of a chromosome. So it depends on the location of the promoter. So according to this one, so transcription, because we have to go from promoter to terminator, so the transcription could be going in this direction, because we are always going to read, again, like DNA polymerase, RNA polymerase is going to read 3 to 5 and make 5 to 3. Okay, so they both work in the same direction. DNA and RNA polymerase, they read 3 to 5 and they make 5 to 3. So we will read 3 to 5 and the RNA is synthesized in the 5 to 3 direction. because it's going to be complementary and antiparallel. Okay? So again, because of the location, particular location of the promoter and terminator signal of this gene, this is going to be the template strand. But for this gene, for instance, okay, this one is going to be the template strand. Again, because of the particular location of the promoter and the terminator. Okay? So it is the location of the promoter and the terminator that dictate which strand to be copied. Okay? But they're always going to be copied in the same way. So we read 3 to 5 and we make 5 to 3. Okay? So I will repeat this. Okay? So the polymerase will locate for gene to be transcribed by searching for the promoter. What is the promoter? It's a DNA sequence to which transcription factors and RNA polymerase bind. So it's the binding of these transcription factor and polymerase to the promoter that will tell us we have to start transcription here, and it will tell us which strand is going to be the template, the one that we are going to transcribe, and what is the direction. Okay? And the transcription will end when we reach the termination signal. So, as I said, the RNA polymerase is going to move along the template in the 3' to 5' direction. So it will read 3 to 5, and it will make 5 to 3 using NTPs, not DNTPs, it's NTPs. Okay, so it's not deoxy, they are all NTPs. And remember, the DNA polymerase had a proofreading, right? Here we don't have a proofreading. It's no big deal because we're just going to make a protein, and abnormal protein will be degraded, but it's not something that we are going to copy to future generation, so that's why no proofreading is required. Right. What about the other strand? We have 2 strands, we said, right? What about the other strand, which is the non-template? The non-template, we call it the coding. Okay, it's a bit confusing. It's non-template. We are not using it. It's not our template, but it's called the coding. Why it's called the coding? I'll tell you in a minute and you will understand. This non-template will be identical in sequence to the RNA molecule that we are making, except that instead of U, we have T. Who can tell me why the non-template is actually identical in sequence to the messenger RNA?
Because it was complementary for the template one.
Sorry?
Because it was complementary for the template.
Yes, because it's complementary and antiparallel. Okay? The two DNA strand. I don't know if I have an example. Yes, okay. Let's look at this one. Okay, so this is my DNA helix. Okay, so my gene is sitting there waiting to be transcribed. My 2 double-stranded DNA, we said it's anti-parallel, right? So they run in opposite direction and they're complementary, right? Once I transcribe, the promoter will tell me, because of the presence of promoter, it's going to tell me that I need to go this way. So I'm always going to be reading 3 to 5. So this is going, the lower band in this case is going to be my template, the one that I will use to make RNA. And when I copy this into messenger RNA, I will read 3 to 5 and make 5 to 3. So it's going to be complementary and antiparallel, the RNA that I'm making, right?
Yes.
And this is how the DNA double helix is. It's complementary and antiparallel. So, at the end, to be exactly the same in sequence like the coding sequence, right? But what is the difference between the coding strand and the RNA strand? Yes, U and T. That's the only difference. Okay? So now we have learned about the promoter. So it is part of the DNA that's not going to be transcribed, but to which the RNA polymerase and transcription factor bind and will tell me where to start. Okay? And we learned about the template strand, which is determined by, again, the promoter. And we know that the RNA is identical in sequence to the coding, the non-template or the anti-template. Okay, now we have some terminology terminology, what is the minus and the plus? By convention, if you want to write a sequence, a gene sequence, if I'm writing a gene sequence and then I tell you at position minus 300, we found a mutation. Okay, that could be a mutation in the promoter region, the non-coding region. طيب, what does the minus mean? It's in the promoter. It's a part that is not going to be transcribed. Okay, it's not used to make protein. So anything that is negative is actually before the transcription site. Sorry, the start of transcription, the start site. So anything that is negative is not actually transcribed. It's part of the gene, but it's not transcribed. I'm not going to use it. You see anything negative here? Did I use it to make RNA? No, it's not going to be transcribed. Therefore, I don't use that information to make protein. طيب, the plus one, who will start site? So plus one indicate where I started the transcription. Anything positive, it means that it is something that has been transcribed. Okay, coding. Okay. So anything that is plus, it means it is the coding region. Anything that is negative is a non-coding region. طيب, what about upstream or downstream? I don't say before or after. So anything that is, to the right, we call it downstream. So anything that is after the promoter, we say it's a downstream region, or in another way of pointing toward the 3'. Okay? So anything that is going toward the 3' is actually downstream. Anything that's pointing toward the 5' is called upstream. Okay? It's like in anatomy. You say caudal and cephalic or something. So like toward the feet or toward the head. So here we have downstream and upstream. Downstream toward the 3', toward the end. ما عجبته لأحمد هاي، ليش ما عجبتك الانالوجي هاي؟ اوكي، أو صاحبك حكى لك شيء ضحكك أو إيش بوشي ما بعرف. Okay, so anything toward the 3' is called downstream. Anything toward the 5' is the upstream. So this is just terminology.
Doctor?
Yes.
What if the DNA promoter region is on the 5 to 3, on the 5 side, the 5'? The RNA, how is it?
Sorry?
If the promoter region is on the 5 strand, from 5 to 3, in the DNA.
I'll show you. Okay, so if you see the first two, okay, so I'm copying the bottom strand, okay? If the last one in gene 3, I'm going to copy the top strand, but in this case, I'm always going to read 3 to 5 and make 5 to 3, okay? So I'm always going, regardless of where is the promoter, okay? The position of the promoter will determine which is the template and which is the non-template. Okay, but I'm always going to read 3 to 5 and make 5 to 3.
So it's going to be upstream and downstream?
No, the upstream or downstream is going to stay the same. It's relative to the one. Okay? No, it's always going to be the same. So, we don't change the terminology here. Okay? So now the template strand, in this case, the template strand is the bottom one, okay? It's the way I write them. I'm always going to write them, 5 to 3, 3 to 5, anti-parallel direction, okay? So I'm always going, so these plus and minus will always be the same, okay? So when you give the numbers, you are going to give them in a way that it doesn't change, okay?
Is the template strand the same as the antisense strand?
Sorry?
Is the template strand the same as the antisense strand?
Template is antisense? I think no, template is a sense.
Sense?
I think so. Sorry? Antisense? Yeah. I don't remember. I used to know sense and antisense. Now I forget it. But you can Google it. Okay. Okay, so now let us review how we are going to copy that information. So we have two double-stranded DNA, okay? The promoter will tell me which one is going to be the template, which one I'm going to copy. When I copy, as I said, I read the 3 to 5 and I make 5 to 3. So my newly synthesized RNA is going to be complementary and anti-parallel to the template. So this is exactly the same as the anti-template or the coding strand. So if you compare the sequence here, look at it. AUG, this is ATG. GGG, GGG. CUC, CTC. So it's exactly the same. Okay, the messenger RNA is exactly the same like the coding strand. And this one, that's what you are going to learn next week. So I'm going to read the code, but I'm going to read the 3 at a time, right? And AUG is going to be the start codon because AUG code for methionine. We learned this in Mol 1. So I'm going to read 3 at a time and I'm going to make the amino acids. And the amino acid is made in a way, the protein is made in a way from the amino end to the carboxy end. So this is the direction at which I make the protein. Type. If we said if we compare the coding, it's exactly the same as the messenger RNA. But when I want to give you the sequence, we talked about sickle cell and we said there is a mutation, right? But when we give the sequence of the gene, we don't give double-stranded sequence, right? We use one strand to give the sequence. Which strand are we going to use to?
Coding.
Yes, exactly. That's why we call it the coding strand. So the one that is not being transcribed, this is not fair, sir? The one that is not transcribed get all the credit and the gene is named after it, okay? Because it's actually the coding. Why it's the coding? لأنه it's exactly the same like the RNA. And because we are going to use that information to make the amino acids. So it makes more sense, because you are going to remember that ATG, it means AUG, so this is going to code for methionine, okay? So when you write the gene sequence, you always use the coding strand to write the gene sequence. You don't use the template strand. You don't use the one that you copy into messenger RNA, okay? The template do all the hard work, it's the one that is copied, and the coding get the credit, the name of the gene and the sequence of the gene. Okay, let's do this question. Okay, we have a DNA template, very small. In the test, I usually give slightly longer. Okay, TAGC. I didn't write 5' to 3', so what do you presume in that case? It is written in the 5 to 3, right? So T is at the 5'. Okay, so now I'm asking you for the RNA sequence. And again, the choices, they don't have 5 to 3, so you need to have the answer written in a 5 to 3, right? So what is the choice?
D.
I can't hear you. You're all saying what? D or E?
D.
Who says E? Why E? Can we write here? Who wants to solve this on the board?
Guys, it's D. It's D. It's D. Wait, what is it? Two. Oh my God.
Like the answer D. Do you agree?
No, C.
What you wrote here is perfect, right? Now you need to write the answer. And when we write the answer, in which direction we write it?
5 to 3.
C, رمض الطرف خطط C.
has to be complementary and to this. So T, A, U, G, C, C, G. Because the complementarity rule. Complementarity. So T will bind to A will bind to T. But you know, in RNA there is no T, so it has to be U, right? Okay, G, C, C, G. So this is how it is, you know, this is the strand. But when I report the answer, I have to write it 5 to 3. Okay, because we didn't specify here. So by convention, when you write any sequence, you write it 5 to 3. So that's why when you write your answer, you write it this way, 5 to 3. So you have to flip it. So you write G, C, U, A, which is answer E. Yes.
Will you tell us that the temperature sequence is 5 to 3? Normally it's 3 to 5, right?
Normally, you know, it's
Yeah, the template is 3 to 5. Yes.
No. So here I'm telling you that this is the template.
Right here.
Okay?
Yeah, but it's a question is not 5 to 3. How do I know?
No. Here, I wrote the template sequence, and I'm telling you, you know, this is, you are going to use this to make RNA. So you just use it to make RNA. The RNA has to be complementary. Okay? You have to flip it. You have to write the complementary base. And when you report the answer, you have to write it 5 to 3. Yes. Yes. Here we are talking about the answer, okay? We read it. So the way the polymerase works, it's going to read it in that direction. Okay? It's going to read 3 to 5 and make the RNA 5 to 6. You know, the sequence that's given here is not the coding sequence. I'm giving you the template, not the coding. By convention, I have to write any sequence 5 to 3. I shouldn't just write it for you. Okay? Yes.
I have a question. The RNA sequence is what?
Can we listen to the question? Yes.
It was about the question that was just given. The RNA sequence is 5 to 3, right? the RNA sequence is written down as 5 to 3.
No, yes, here the choices, they are written 5 to 3.
Yeah.
Okay, when you write, here I'm not talking about the direction, I'm not talking about anything, but when you write in an exam or in any paper, you see a sequence, you have to write the DNA or RNA sequence in the 5 to 3 direction.
Okay. The DNA template given, is it in the 3 to 5 prime direction or the 5 prime to 3 prime direction?
No, here forget about the direction, okay? Forget about the direction. I'm giving you a small sequence and I'm telling you what will be the RNA made from that, okay? That's it. So this is, I'm writing the sequence by convention. You have to write it 5 to 3. This is how you write any sequence, okay? It's not here you are the polymerase and you are reading the sequence. No. When you write things, this is how we write them. Always 5 to 3, okay? Yes. Yes, Anis.
Doctor, in the question you said TAGC.
Yes.
Is this sequence 3 to 5 or 5 to 3? It was not mentioned. But you said in the beginning that this is 3 to 5.
If you see a sequence, and I don't tell you what is I don't write the direction 5 to 3, you have to know that it is written in a 5 to 3. So if you don't point to the direction, I've been saying this from last week. If I give you any DNA sequence and I don't indicate 5 to 3, you have to presume that I'm writing it 5 to 3. Okay, so this is very important. Yes? Can I hear the question because I have to go? مين عندكم هلا؟ مين؟ علي. And we can take the attendance. Let's take the attendance.